Hello everyone, this is vijaya Lakshmi studying B Pharmacy first year in Maharaja College of Pharmacy. So today I am going to discuss about a topic called electron transport chain. So before going to this topic, we'll discuss about what is biological oxidation. So biological oxidation is nothing but some energy rich compounds such as carbohydrates, fatty acids and amino acids, so they undergo a series of metabolic reactions and they produce some reducing equivalents and that are oxidized to CO2 and water. So these reducing equivalents are transferred to the coenzymes such as NAD and FAD and then are converted into NADH2 and FADH2. So these coenzymes passes through the electron transport chain and results in the liberation of some free energy, and that free energy is important or utilized in the synthesis of ATP, so that we are calling as oxidative phosphorylation. So we can conclude that in biological oxidation includes both electron transport chain and oxidative phosphorylation. So electron transport chain is nothing but the transfer of electrons, that means the energy from the substrate, and results in the and finally it results in the formation of water. And oxidative phosphorylation is nothing but the synthesis of ATP utilizing the free energy which is liberated in the electron transport chain. So now coming to the electron transport chain. So electron transport chain definition. So electron transport chain is a series of protein complexes that couple redox reactions, creating an electrochemical gradient that leads to the creation of ATP. So electron transport chain, simply we can say that, so in the electron transport chain includes 5 complexes, so that complexes are important for the transfer of electrons in that chain and results in the formation of a electrochemical gradient was formed, and that electrochemical gradient is important for the oxidative phosphorylation, that is ATP synthesis. So now coming to the, so where the electron transport chain and the oxidative phosphorylation are taking place means, so the location is inner mitochondrial membrane. So both the electron transport chain and oxidative phosphorylation taking place at the inner mitochondrial membrane. So we'll briefly discuss what is electron transport chain. So we'll discuss biological oxidation. So here we can see that in the first diagram, in the overview of biological oxidation, that is under electron transport chain. So here, the as I already said, that carbohydrates, fatty acids and amino acids, they undergo a series of metabolic reactions and then finally oxidize to carbon dioxide and water, and during this metabolic reaction some reducing equivalents are formed, so that are catched up by NAD and FAD and that are converted into NADH2 and FADH2. So these NADH2 and FADH2 are the coenzymes that are transferring into the electron transport chain and giving rise to and reduced oxygen to water, and during this electron transport chain some free energy is liberated. As I already said, that free energy is important for the conversion of ADP and inorganic phosphate to ATP, that means the synthesis of ATP. So now coming to the overview of electron transport chain. So as I already said that electron transport chain means the transfer of electrons from the substrate. Here under the overview of electron transport chain we can observe that, so here A is nothing but the substrate. So here the substrate undergo, for example we will take carbohydrates as substrate, so they undergo series of metabolic reactions and they will liberate some reducing equivalents. So that reducing equivalents are transferred to the coenzymes. So here we can see the coenzymes NAD, and that are converted to NADH2 and that accepts the proton, and NADH2 is converted to NADH2 and that are accepted by the flavoproteins, and that flavoproteins accepts the protons and gets converted to FPH2. And so here also there is a liberation of the proton, and these are then the energy is transferred to the cytochromes, that are cytochromes and gets converted and it is reduced to water. And this is about the electron transport chain overview. So we can go with the mitochondria. So why we are discussing about mitochondria means, as I already said that the electron transport chain and oxidative phosphorylation both will take place at the inner mitochondrial membrane of the mitochondria. So we'll just look into what is mitochondria. So we already know that mitochondria is the powerhouse of the cell, and mitochondria is the centers for many metabolic oxidative reactions to generate reduced coenzymes NADH2 and FADH2, which in turn are utilized in the electron transport chain to liberate energy in the form of ATP. So in the inner mitochondrial membrane, both the electron transport chain and oxidative phosphorylation are taking place. So this will result in the liberation of some free energy, and that free energy is important for the formation of ATP, that is synthesis of ATP, that is we are calling as oxidative phosphorylation. So we'll take a look into the mitochondrial organization. So how the mitochondria is organized as a parts. So mitochondria is distributed into 5 distinct parts. So mitochondria is divided into 5 parts, that are outer membrane, inner membrane, intermembrane space, cristae and matrix. So the outer layer of the mitochondria is known as outer membrane, and the inner layer is known as inner membrane, and the space between these two layers is known as the intermembrane space. And the mitochondria is highly coiled, and that is called cristae, and the space present in between the inner membrane, that is called matrix. So here inner mitochondrial membrane is impermeable to ions like H+, K+, and Na+, and some small molecules such as ADP and ATP. So in the structure, the inner mitochondrial membrane is containing some phosphorylating subunits. So they are the small subunits which are important, or where the ATP synthesis is taking place, and we can observe the F0 and F1 subunits. So here where the actual ATP synthesis is taking place. So for the synthesis of ATP, so as I already said that inner mitochondrial membrane is impermeable to H+, K+, and sodium, and ATP also. So though the inner mitochondrial membrane is impermeable to ATP and ADP, so that's why there is a generation of electrochemical gradient. So by the generation of this electrochemical gradient, so it will lead to the formation of the ATP. So electron transport chain is coupled with the electrochemical gradients, which will result in the synthesis of ATP. So now coming to the electron transport chain with enzyme complexes. So the electron transport chain, so as I already said that electron transport chain is taking place at the inner mitochondrial membrane. So at that place the enzyme complexes are present. So inner mitochondrial membrane is having 5 enzyme complexes. So in that first 4 enzyme complexes are involved in the electron transport chain, and the later one, that is the 5th enzyme complex, is important for the oxidative phosphorylation, that means the ATP synthesis. So here we can see that first 4 complexes are responsible for the transfer of electrons, that means the electron transport chain, and the 5th enzyme complexes is important for the ATP synthesis, that means the oxidative phosphorylation. So here we can observe these enzyme complexes. So the transport of electrons from substrates combines with H+ and oxygen to produce water. So as I already said that the electron transport chain is taking place from the, or precursor is the substrates, and that will combine with the hydrogens, that are protons, and that will result in the reduction of reduces to water and CO2, and it will produces under substrate undergoing metabolic reactions, and it will results in the formation of, it will giving some protons, and that protons are accepted by the NAD and results in the formation of NADH2, and that protons are transferring to the FMN and FMNH2, and it will result in the formation of FMNH2, and it will give electrons are transferring to FES, that are FES is nothing but the iron sulfur protein. So that energy is transferred to the iron sulfur proteins. So iron sulfur proteins are exist in 2 forms, in oxidized and reduced form. So in oxidized form, iron sulfur proteins are exist in Fe3+ form. In reduced form, it is exist in the Fe2+ form. So here the electrons are transferring from the FMNH to FES, that is iron sulfur proteins. So here the complex 1 is present. So the complex 1 is NADH CoQ reductase. CoQ is nothing but the coenzyme. Q is an lipophilic electron carrier, so it can accept electron from the FeDH2, which is present outside the electron transport chain, and it can accept electron from the FeDH2 in the electron transport chain. So coenzyme Q is absent in myobacteria. So in the place of coenzyme Q, vitamin K performs similar function of the coenzyme Q, and it will result in the, or it will perform the electron transport chain. So here you can observe that from the succinate. So here that is the substrate. So from that the electrons are transferring from the FAD, and that is converted to FADH2, and that electrons are transferred to FES, that is iron sulfur protein. So here the complex 2 is present, that is the succinate and CoQ reductase. So this is the second enzyme. And then coming to the coenzyme Q, from the coenzyme Q the electrons are transferred to the cytochromes. So cytochromes are nothing but these are also the proteins. So these are the small proteins. So they are divided into cytochrome B, C and A. So the cytochromes. So cytochromes are nothing but these are the conjugated proteins that are containing in heme group. So cytochromes are divided into A, B and C type. Cytochrome C contains 104 amino acids and a heme group, and the cytochrome A and 3 is also known as cytochrome oxidase. So we already said that cytochrome A and A3 are the terminal components in the electron transport chain. So here the electron transport chain ended with cytochrome A and 3, and they are collectively known as cytochrome oxidase. And from this here the complex 3 is present, that means present between the coenzyme Q and cytochrome C reductase. Why we are calling as this enzyme means the electron transferring from coenzyme Q to the cytochromes, and that means the reduction is taking place, and then the electrons are transferring from the cytochrome C1 to the cytochrome C, and then to cytochrome A, and then to cytochrome A3. And so here the complex 4 is present in between the cytochrome A and A3. So as I already said that the cytochrome A and A3 is also collectively known as cytochrome oxidase. So and that is the terminal component in the electron transport chain. So here the oxidation is taking place, that means the addition of oxygen at this complex, and it will result in the formation of water, and it will ends the electron transport chain. So here from all these complexes, all the 4 complexes are involved in the electron transport chain. So as I already said that the 5th NJ complex is important for the ATP synthesis, that means oxidative phosphorylation. So the 5th complex is containing an enzyme is known as ATP synthase, which is important for the oxidative phosphorylation. So as I already said that through the electron transport chain some free energy is liberating, and that free energy is important for the conversion of ADP and inorganic phosphate to ATP, that means the synthesis of ATP, which is taking place at F1 and F1 subunits of the inner mitochondrial membrane of the mitochondria. So the complex 5 is important for the oxidative phosphorylation. So this is about the electron transport chain and oxidative phosphorylation in brief. So the transport of electrons from substrate combine with hydrogen and oxygen to produce water. So as I already said that in the electron transport chain, the finally reduce it to oxygen and water, and it produces the water. Electron transport chain can be concluded by this way. So at the end of the electron transport chain, electrons are transferred to molecular oxygen, which splits in half and takes up H plus ions to form water. So as I already said that in the electron transport chain, the electrons are transferring and finally combines with water and split, and the water is splits in half, that means it is in the formation of half O2, and this oxygen, molecular oxygen, will takes up the H plus ions, that means the protons, it is results in the formation of water. So this electron transport chain is coupling with the oxidative phosphorylation, that means next to the electron transport chain, oxidative phosphorylation is taking place. By the liberation of some free energy, this oxidative phosphorylation takes place, that means the synthesis of ATP. So this both will comes under the biological oxidation, that means the electron transport chain is collectively with the oxidative phosphorylation is known as the biological oxidation. So for this I referred this topic from U Satyanarayana and U Chakrapani book. Thank you.